They're going to feel like I'm yelling at them the whole time. There's no trash can in here. I know, right? I think we have some vacation ones. Your dad. He converted them all. Ben's dad used to have all these slides, and every time we would go visit them, he would take out all the slides and show us all the slides over and over again. Probably not. Like Tarzan. Yes, like Tarzan. Or what?
Now you're beginning to sound like my daughter.
All right, so I want you guys to get involved more today. So what I've done is I've tried to come up with a case study that should be really interesting to you. At least I hope it's going to be interesting to you. Brad, I'm sorry, there's no math. So for those of you who don't know, the reason why I always pick on Brad is, number one, Brad's worked for me for a long time. And number two, Brad has his bachelor's in electrical engineering and a master's in mechanical engineering, and now he's starting a PhD in biomedical engineering. So he's an engineer at heart who's trying to be a biologist successfully. There's a little bit of math. Yep, a little bit of math for you. So today we're going to talk about enzymes. And like I said, there's some great case studies here because as we talked about yesterday, enzymes is where a lot of drugs are targeting. So first question, have you or anyone you know ever had an adverse drug reaction? I'm seeing heads nodding yes. Does anybody want to share?
Of course, you don't have to.
No, you don't have to, but yeah. Did you? Yeah, a penicillin reaction. Those are really common. Yeah, of course you don't have to share, but if you want to share, you're welcome to. So we actually can do a lot now to help prevent drug reactions. And one of the things that we can do is genetics. And so this has spawned this whole new field called pharmacogenetics. And one of the things that we specifically look at are people who have different ways of processing drugs. So they metabolize the drugs differently. And in this particular case, this is what that type of report looks like. So antidepressants are really, I mean, we all know how many people are on antidepressants. It's a huge portion of the population. Antidepressants are something that get metabolized very differently depending on specific enzymes that you have. And that goes back to your genetics. So in this particular case, this tells us a list of different antidepressants, and then it tells us if there are known gene-drug interactions. So is this going to interact with any genes that we know of? It tells us if there's going to be any options clinically indicated, and CYP450 is going to be a tremendous part of today's lecture. And you will hear this probably in every class you ever take in pharmacy school, because CYP450 is one of our main metabolizing enzymes that we have. So we're going to talk a lot about CYP450. Have any of you guys ever heard of CYP450? Cool. Cytochrome P450. All right. Well, you'll learn a lot about it today, hopefully. So this is one of our major metabolizing enzymes. And what we figured out is that if CYP450, it has a lot of different genetic variabilities to it. And with certain genetics, it doesn't work well with certain drugs. So for this particular patient, and this actually came from a particular patient, this tells us that this particular drug, it's not one of us. No, this is something I pulled off online because somebody is posting something. It's de-identified, so it's not covered by HIPAA. Anyway, so you can see that, like Prozac here, really common antidepressant, right? Well, if you look across here, okay, enough out of you. So Prozac, you can see that if this person had this particular mutation in SLC6A4, it will likely cause an adverse response for that particular individual. Well, Prozac is one of the first drugs that a doctor would try. So now we've put somebody on Prozac who, if we took the time to look at their genetics, we would see that they're likely to have an adverse response with Prozac. But we could choose Cymbalta, and they would be fine. This is the power of pharmacogenetics. So today we're going to talk a little bit about how we can use some of the information we can get, and we've talked a little bit about this in the past, but what this means in terms of enzymes and how enzymes function. Okay, next case study. Mr. Jones is a 40-year-old man. He weighs 230 pounds. He's taking Seldane, which is basically an antihistamine. This is not Ben. So he got sick with bronchitis, and he was given erythromycin, an antibiotic. Shortly thereafter, Mr. Jones was treated at the ER for chest pains and was diagnosed with a cardiac arrhythmia characterized by altered cardiac repolarization and prolonged QT intervals. He has never had any cardiac trouble previously, and his blood glucose is normal. What happened? What happened to Mr. Jones that he now has a cardiac arrhythmia? You're right. It's an adverse drug interaction. So do you guys know how to look up adverse drug reactions? You can actually go to the FDA website, and there's also a website called drugs.com, and you can put in 2 different drugs and look for their interactions. That's going to be something that you're probably going to want to do, and I don't think we have time for me to walk you through that today, and it's not on your test, but it's a pretty cool thing to try to mess around with to find some of these adverse drug reactions. So it all goes to cytochrome P450. So the cytochromes are also known as CYPs. These are enzymes that catalyze. Yeah, it's a big thing, and we'll talk a lot about this in pharmaceutical calculations.
Yeah, and they, it's actually one of the leading causes of death in the United States.
So that
Over 100,000 people die.
that particular drug interaction killed 8 people before they pulled it off the market. They pulled the antihistamine. And it's a really fascinating story, so that's why you're going to learn, hopefully, and get really interested in this story. So cytochrome P450 is an oxidation reduction reaction. It metabolizes approximately 60% of your prescription drugs. CYP450, 60% of your prescription drugs. So that's the cytochrome, sorry. So then CYP450 is a superfamily within that, okay? And it's called 450 because it has a peak absorbance at 450 nanometers, sorry. Then in this family, there is a subfamily, and this particular participant is CYP3A4. And CYP3A4 is about 34% of all the cytochromes, CYP3A4. So it's found in the liver and intestine. This is what means that grapefruit juice can interfere with some of your drugs, is CYP3A4. There you go. So it turns out that, and you'll learn this in pharmacokinetics, that it takes quite a bit of grapefruit juice to interfere with it, but if you get enough, that will hang around for 3 to 7 days, and your drugs won't work, or they'll work too well, and you'll overdose. So that's why this is a really important topic. But you can see that CYP3A4, and they group 5 and 7 in here because they're hard to distinguish, is about 30%. And then you can see all these other members of this family that are going out here. Here's the crystal structure of CYP3A4. Ben is going to show you that here in a little bit. It's a hemoprotein meaning that it has a heme group. It oxidizes small foreign molecules, and you can see that there are substrates for it, so things that it works on, and there are inhibitors and inducers. So inducers make it work better. Inhibitors are going to stop its action. And you can see that, like, omeprazole, do you guys know what omeprazole is? Right? Yeah, we see a lot of people on omeprazole. That is hitting CYP1A2, and that is inducing it. Phenobarbital is hitting CYP3A4, so that's seizure meds, right? So if your CYP3A4 isn't working well, your seizure meds may not work well. Okay, so now let's look at Seldane. Now this is where the story gets really interesting. So Seldane, here's your histamine, right? That's what you're actually reacting to when you have an allergic reaction, is histamine. So in purple, you can see the outline of the pharmacophore. Now remember, the pharmacophore is where that base of action is. All the other stuff is just kind of what they're doing to make it stabilized, or they have to have those functional groups there to produce it. But the pharmacophore is what gives it its action. So the pharmacophore of histamine is the same as the pharmacophore of Benadryl. And Benadryl, though, crosses the blood-brain barrier, and it makes people tired. Have you guys ever taken Benadryl? Yeah. Does it make you tired? Yeah, it makes a lot of people really tired. In fact, when babies don't sleep, they say, just give them a little Benadryl. When dogs don't sleep, give them a little Benadryl. It's all good. That's the wrong class. Anyway. Okay, so then when you look at Seldane, this is Seldane right here. Here's the pharmacophore, so it's the same. But X over here is a methyl group. What do we know about methyl groups? Are they polar? No. Do they have a lot of interactions? No. They're very, very stable. But it turns out, and I don't understand this chemistry, I actually talked to several chemists today, and they're like, how is this happening? And I went, I don't know. I don't know how this is happening, but this is the coolest part of the story. So if there is a methyl group right here, you get heart toxicity. If there's a methyl group there. So you're like, well, gosh, why would they make something that they know can produce heart toxicity? Well, it's because Seldane gets metabolized by CYP3A4. And CYP3A4 undergoes this oxidation. It helps with this oxidation reaction. And you convert this methyl group into COOH. And that's Allegra. And now Allegra is perfectly safe. But what happened was, well, before we get to me telling you what happened, this is Seldane. Here's Benadryl, and here's histamine. These are electrostatic plots of them, and you can see some of the reason why all three of these work the same is because of where the electric charges lie, which I think is really cool. So it's not only structural, but it also has to do with electrostatic interactions. Okay, so CYP3A4 can be inhibited by erythromycin. But CYP3A4 is completely necessary to convert Seldane, the heart-toxic drug, into Allegra, the heart-safe drug. So now if we inhibit 3A4, we increase the blood level of whatever drugs CYP3A4 is supposed to be processing. So when they gave erythromycin to Mr. Jones, and he's on this antihistamine, what they ended up doing was blocking the ability of CYP3A4 to convert this methyl group to COOH. So they blocked the conversion of Seldane to Allegra, and he had a heart problem. It actually, this is the effect that killed 8 people and caused Seldane to be pulled from the market. Because they're like, well, hey, if Allegra works, just sell Allegra. So now that's why we just have Allegra and we don't have Seldane. There are other things that can induce CYP3A4 to work better, which increases your clearance and decreases your blood levels. So we have to understand what CYP3A4 is doing with the different drugs that we give. That's a cool story, you guys. I know you should be impressed. I got really excited when I got to research this story today. Yeah? Absolutely. Yeah. Well, okay, except for it was given anyway. And the reason being that when they did the clinical trial, they did the clinical trial with people who were not taking any other medications. So they didn't think about how Seldane was metabolized to make it convert from this heart toxic drug to a safe drug. And if they had thought about it, they would have realized that if they gave any other drug that blocked CYP3A4, then they were going to have a problem, because then the cytotoxic level builds up of Seldane and you get the toxic heart effects. So, yes. Yep. But now, I mean, and that's when they pulled the drug. But that's the reason why it's important to mention, right? Because grapefruit juice interacts with statins, right? Statins are cholesterol-lowering medications. Lots of people are on statins. So it's important to know, and a lot of times patients don't disclose all these things to their doctor because they're like, well, I'm taking over-the-counter acetaminophen. Acetaminophen gets processed, right? Or they drink a grapefruit juice every morning. And it turns out that it also happens with pomegranate juice. So they don't mention that, because why would you mention that, right? You wouldn't think.
Pomegranate juice.
Lots of people drink pomegranate juice. Anyway, so why would you mention that? But as pharmacists, you're going to have a very different relationship with your patients, right? You're probably going to have a much more intimate relationship with your patients than the doctor does, because you're going to see them once a month when they come to pick up your medications. You'll know Mr. Jones. They'll say, hey, Mr. Jones, they prescribed you erythromycin, but I noticed that you've been picking up this antihistamine. And it also happens with antifungal drugs a lot. So I noticed, yeah. And carbamazepine is like a psychotic, right? And so you will know that drug history. The doctor won't necessarily think to ask all those questions and think about the diet and think about all the over-the-counter meds that they're taking. And sometimes people don't want to disclose those things to their doctor. They don't want their doctor knowing that they're taking this herbal supplement and they're taking this because they're trying to manage their health on their own, right? So they don't necessarily disclose that. But as a pharmacist, you guys are safe, right? It's a safe space, and they'll talk to you about things that they may not talk to their doctor about, which is why it's important that you're aware that these enzymes, these drug targets, can be attacked and affected by multiple drugs. And sometimes those drugs have different effects on that same enzyme. That's what leads to drug-drug interactions. Oftentimes, anyway. So what are enzymes? Essentially, enzymes are catalysts, right? We talked about that yesterday, where catalysts are the name that they call enzymes in chemistry. But enzymes are catalysts. They increase the rate of reaction, but they don't get used up, which makes them very unique. And they're not permanently changed. Actually, Ben, I'm going to have you do your Pymol with CYP3A4 so that we can finish up that story here quickly before we get into this. Okay, so Ben's going to show you CYP3A4 because it is really an interesting molecule. And then we'll go through the different classifications of enzymes. You're going to see a couple different Pymols today. And I think Ben is going to try to do some really cool things where he's going to actually move molecules into and out of binding pockets. So hopefully you'll be able to see how these things actually fit. Did you do erythromycin and CYP3A4? How erythromycin actually impacts CYP3A4. And then we'll get started on enzymes. Any questions while he's finishing setting up about what exactly we're trying to do?
Wow.
It always freaks me out when you do that. I think it was the early 2000s when it got pulled from the market. I want to say 2004. Well, so it's a good question. So current drug trials, they start out with phase 0. So it depends on the phase of drug trial that you're in. At first, you're just looking for safety and efficacy, and they're looking for drug reactions and drug interactions. But typically, the patients that they're going to enroll are going to not be taking anything. But even on those clinical trials, now they know to ask if you drink grapefruit juice or various things like that, but they may not know to look for everything, because we don't know what we don't know until we don't know it. So, and then it continues to progress. So you start with safety and efficacy, and then you continue to progress to look at even additional safety and efficacy. And it's a big problem with clinical trial designs in terms of what actually is the contraindications, meaning we're going to exclude people who are on this particular drug. We're going to include people on this drug. So we're actually doing one for looking at some obesity patients and looking at the microbiome of obesity patients. And we have to decide whether or not we want to exclude people who have type 2 diabetes, because they're going to be taking different medications, right? But if we exclude them, then we're going to exclude a large chunk of the obese population, and so we may not, it may be too restrictive. So this is the problem that you have when you design clinical trials, is trying to figure out who do you include and who do you exclude. Yes. Yep, and so then it all goes to the labeling then, and you say this is labeled for this particular indication in this group. There's an interesting story that came out on 60 Minutes a couple years ago where they looked at clinical study designs, and they found that most clinical study designs from early on through even 1990s included more men than women, which is why there's this huge push to have equal representation from all these different groups, because it turns out women process drugs very differently than men process drugs. And so drugs that are safe for a man are not necessarily as safe for women. And this has been a large issue that they're trying to figure out what to do with. And do they make people go back and redo all these clinical studies? Because this is millions and billions of dollars worth of research. So it's an interesting quandary about what do we do, because maybe we didn't do the best clinical study designs, but the drug is safe for the majority. So yeah, it's thresholds, exclusion, inclusion. It's all pretty fun.
Okay, so we're going to talk about Cytochrome P450 here. I just did a simple Google search. This is a molecule of the month. At this point, hopefully you've at least looked up that website and have an idea what I'm talking about. This one has definitely excellent graphics. We're going to go down to this model here, 2JOD, and look at it. It's the one with the erythromycin on it. So it's this right here, and you can see erythromycin sticking into that. So Cytochrome P450 has chains A and B, and we'll look at that in a second. There's a heme molecule. You'd be surprised how many things have heme in them. It's kind of actually quite interesting. And then there's erythromycin, and so erythromycin's ERY. If you want to get into the ligand view, as noted earlier, you can pull this up and zoom in and see how the erythromycin actually interacts and what residues it interacts with. So you can see it interacts with this alanine, this tyrosine, this threonine, alanine, phenylalanine. You can see exactly where and you can also go down and see what kind of bonds it interacts with. So you can see there's hydrogen bonds in there that are blue. So we'll do a PyMol window. Fetch 2J0D. And this is a dimer, but it's not really a dimer. And you can see how, so see where the heme group is? Just hide everything here. Then we're going to do select SIP, comma, chain A. And we'll just show as a cartoon. Go up to display. I don't know how they can't fix that bug. It drives me bonkers every single time. Okay, so then we're gonna color this. I'm gonna color it gray just to give you. You can see where that erythromycin sits in there. So we'll make that, show it as a sphere. Color it red to give it some contrast, and you can see exactly how it fits in there. If we go to this as a surface, you can see the binding pocket very nicely and how it fits kind of right in that spot to deal with. All right. So if we want to make a quick movie out of this, there's a website. No. So if you go to PyMol movie, definitely do not want LOL movie. Movie School, there's a PyMol wiki about it, and it basically takes you through all of the steps about making a movie, introductions, blah, blah, blah. If you are in Tegrity, oh, did I move that over? Maybe I didn't. Maybe I haven't moved those over. I forgot about. Have you guys watched the videos? Do you guys know what I'm talking about? PyMol tutorials? Well, someone should have said something. All right. I'll move those over just so that you can listen to me drone on and on and on and on about PyMol. It's really exciting. If you go to Integrity class, and I'll move these over right now so that you can see them. I don't know. So there's PyMol tutorial number 1, PyMol tutorial number 2, PyMol tutorial number 3, movie making. Those are all really good videos, and I'll just move. Oh, yeah, I guess I can't since it's doing that. But anyway, watch PyMol. So if you want to know how to make movies, this is where you can do it. So you're going to type in, both those are resources. You're going to type in mplay. We're going to go to scene, and we're going to store F1. Then we're going to right-click here, drag, shift, middle mouse button, move this over here. And this actually may go backwards, but then we're going to go to scene, store F2, and then we'll go to movie, program. Let's go to YROC, 30 degrees over 8 seconds. That was really bad, but you know, I didn't actually make it do what I wanted it to do, but that's generally how you do actually make it work. in the video, so I would encourage you to watch the video instead of just, I just screwed that up big time, but that's the general idea. Is you make scenes, and then you can make it zoom in and go exactly where you want it to go. It just saves the main screen. And then you can just go here to File, Export Movie as MPEG. And it'll just, it'll save all of that out as a movie. And then you can just incorporate it into whatever movie you're making. It takes a little while. Probably take, this one says a couple minutes. So that's.
How would they make the movies with 3D process? Do they just import that movie into?
Yeah, so you could, if you like, say you're doing it, yeah, you can do PowerPoint, you can do Movie Maker, you can do Windows Movie Maker. The most popular is the one on the Mac, the Movie Maker one on the Mac, whatever it is. iMovie, thank you. And then PowerPoint's really easy. A lot of times I use that. You can also do Tegrity. A lot of people use Tegrity. The thing about Tegrity is you can get a lot of help from the help desk from it. In fact, you don't have to know anything. And you can just sit there and record your computer and you can talk into it. And then you can go and edit and cut out certain frames as transitions. And so it's virtually seamless for if you wanted to record. I have a movie on how to make a movie. Okay. So go to slideshow, record slideshow, blah, 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 you talk, file, export, MP4. Then you upload it up to Tegrity. Now, I would not encourage you doing it on a Mac. For some reason, in the past year, something has happened with Mac PowerPoint that it does not upload to Tegrity correctly. You can record it, then you save it, and you put it on a PC, so you can go to the lab or whatever, and then export that out, and it works just fine. I used to record everything on my Mac all the time, all the videos that you pre-lecture videos. I used to record those all the time on the Mac, and then sometime between this year and last year, it stopped working. So now I have to actually go into my Windows and save it in PowerPoint on Windows and export it out. No, it is not just me. All right, so that's the structure of Cytochrome P450, and since it's doing this, it'll probably take a while, so I'll just let you. Yeah. Yeah, because I did hide one of the chains. You did, oh, okay, so one thing also happens, did you type 2J O or 0? 0D. That's the right one. Actually, you can come see me after class, and I'll go over it. Well, maybe not after, maybe before class night tomorrow. Come see me, and I'll show you. Yeah. So you could type in here, select, space, whatever you want to name it, Billy Bob, comma, chain. It is important you put capital. So whatever the B. So that's why looking at the PDB is important. Then that will put Billy Bob over here, right? And then you can edit it. You can do all the actions against it. You can show it in the different forms. You can hide it. You can color it. All the fun stuff. Yeah. Yep. So it's a great question. And that's actually a hole in the mesh. Usually you see that where two molecules interact. I'm not certain why you see a hole in that mesh right there. So if you look up here, you can see there's certain ones that aren't actually colored and highlighted. They don't see those in the crystal structure. Those are missing. That's a hole in the mesh. That's probably not the right term for it, but does that make sense? So that there's this leucine, arginine, phenylalanine. That whole stretch is just missing out of the molecule. So one of the things that I did, and that we commonly do, so we don't know the crystal structure of SOD1A for I'll show you this here in a minute, but we don't know the crystal structure for SOD1A. So, but we can use a computer to predict what it is. So, but I'm getting ahead of myself.
Okay, so now you guys have seen CYP4A, 3A4, I guess. So now let's talk enzymes. So we already know what enzymes are. We saw yesterday how they actually act on the activation energy, and we'll dive a little bit more into that today. Remember that most enzymes are going to be these globular proteins, so they're going to have tertiary structure. Some of them are even going to have quaternary structure if they have multiple subunits. So remember, they're mostly globular, which means if they're globular, what kind of residues are going to be on the inside? Hydrophobic, mostly. Yep, good. And then there's also some RNA or ribozymes that can catalyze reactions. And we're not really going to deal too much with those. Just know that they exist. So as Ben pointed out yesterday, there are certain reactions that take a really, really long time. And so that impacts what kind of enzymes we use. This particular chart tells you how long does that enzyme last. So enzymes, just like drugs, have half-lives. This is what you're looking at right here, is this half-life. So we can see that carbonic anhydrase, which we'll look at later, that's a pretty cool story too, 600,000 seconds before it turns over. Lysozyme, which does exactly what you think it would do. It lyses cells open. Lysozyme turns over every half a second. So this tells you how fast those enzymes turn over. That's going to impact how quickly that reaction can happen in some cases. So we touched briefly on these protein-protein interactions. Remember that you've got enzymes, and then you have protein-protein interactions, and they work a little bit differently. So protein-protein interactions are generally flat and large surfaces, and so you're looking at surface area interactions. But enzymes usually have cavities. They have active sites. They have places, they have binding pockets. They have places where drugs can go and fit in. They have places where
This is an enzyme right here. And if you look at that long enough, you can see exactly where that cavity is.
so you can tell exactly where that drug should bind. And that is really quickening how many drugs we can discover. Because we can use computer programs now. We see where the binding pockets are, and we can say, oh, well, if I change this residue, that changes the shape of this binding pocket, that enzyme may not work. And so we can predict drug discovery based on these enzyme maps. So So you're seeing this over here.
You're seeing a lot of that. So things like your Remicade, your Humira, your Rituximab,
fall over here.
Your things that you probably that are traditional, what you're used to thinking of, small molecules, your aspirin, penicillin, your statins.
A good chunk of the drugs.
Your drugs right now
Yeah.
are drugs that are small molecules that fit in those receptors. Those are very, very different types of drugs. And you can see that based on the interfaces that you're associating.
So remember,
Most of the new drugs that you're seeing are large antibody-based protein-protein interaction drugs.
so remember those are two different types of interactions. Protein-protein interactions occur kind of on that surface or that face level, whereas enzyme receptor interactions occur usually in a binding pocket or an active site. So keep in mind, this is what enzymes do. Enzymes act as blank in cellular reactions. So what do they act as? There's a key word here that you should know. Catalysts, yeah. So remember, here's our starting, here's our ending. We've got our delta G. Remember, delta G only cares, that's Gibbs free energy, it only cares about where you start and where you end. It doesn't care about the path it takes to get there. Enzymes act on the rate. They don't change the energy level of your starting and your ending. They only act on the rate at which it takes to get there. So they're going to lower your transition state. Again, they don't affect the equilibrium. They don't change if you're going to have more reactants or more products. They only change the rate. So because slow reactions face this kind of massive delta G double dagger, so that activation energy, enzymes are going to come in and they're going to help lower that so that the reaction can proceed. And enzymes typically decrease reaction rates, which are called K, by decreasing that activation energy. And here's the equation that shows you how to do that. This is not really an equation that we're going to use that much. We're going to use other equations. So again, a lot of these physiological reactions couldn't really take place under a time scale that is relevant, is physiologically relevant. So we can see that there are certain reactions up here that this is the non-enzymatic half-life. So this is the uncatalyzed rate of reaction, which means that it's not really going to occur. But there are certain reactions that we absolutely have to have occur. And so we can get this rate enhancement, or you can see how the rate changes once it gets catalyzed. So it's this massive change, and that's what enzymes do. They catalyze these reactions so that they can occur on a scale that matters to us. There are 4 factors that I want you to think about that affect how enzymes function. There's your temperature at which your enzyme is active, the pH of the surrounding solution. We saw that yesterday, where pH makes a big difference. There are cofactors and coenzymes that are sometimes necessary. And then occasionally we'll see inhibitors. Those are 4 factors that can influence how that enzyme acts. So a lot of enzymes require a cofactor. Now a cofactor is specifically just a molecule that's necessary for that enzyme to act. A lot of times it's going to open up that active site, or it's going to structurally change that enzyme in some way. There are essentially 4 cofactors that you need to know, and you need to have an example of an enzyme that needs those 4 cofactors. Because you might get a question that says, all of the following are cofactors except for. These are the essentially 4 groups or 8 cofactors you need to know. So FAD, NAD+, those are going to be incredibly important when we talk about glycolysis. Your coenzyme A or CoA, your biotin, zinc, magnesium, manganese, and potassium. Manganese we're going to see is very important when we talk about superoxide dismutase, which Ben will talk about in a couple minutes. Actually, right now. How was that for transition?
So one thing, you'll see that enzyme cofactor question and discussion point in Unit 7 pretty heavily. And we'll add 1 cofactor in there in the discussion. Actually, it'll be ATP. And there's also CoQ in there, but we don't really have that.
There's a couple more, but those, if you start out by knowing those 8 and the enzymes that are associated with it on that chart, you'll be in good shape.
All right, so we've talked a lot about already, and that's what this enzyme is. We've talked a lot already about SOD1. This enzyme right here is SOD1, and I'm going to show you a couple of things that are unique to SOD1. SOD1 is does have the requirement of requiring a cofactor, but SOD1 basically takes superoxides and makes them less superoxide-like. They are still oxides, but they're peroxide, which has a short half-life and can then function in a cell. So I'm going to show you something related to more of your video than I am necessarily with the enzymatic function. This enzyme actually is a dimer. When you actually look, when you pull it up into Pymol, it is a dimer, and the two are necessary to form that pocket.
Do you guys understand what a dimer is? Yeah, you basically have 2 units of it. And those units are typically identical.
We don't have a crystal structure for canine SOD1A. So what I did is what's called homology modeling. So when you get to doing your project, and if you're like, hey, I can't find a crystal structure for, because this is important to me, it's important to her. Our dog has this problem. 2 of our dogs have this problem. The active site actually works just fine. I'll show you this here in a minute. The active site of the enzyme works just fine. It's actually a disease where and this is going to be on your test, so you want to pay attention. It's a disease where, because ALS in humans also affects this, and when we talk about therapies, thinking about different ways that therapies can be delivered is what this is about. So there's at position 40, there is a mutation. Anyway, I'm getting ahead of myself. So we don't have a structure of this, okay? So I did what's called homology modeling. I went to, if you get stuck in a situation, I went to this Swiss model. We do have the human version of this, which is going to be very similar. So Swiss model will go and it'll predict what the structure looks like based on the structure you give it. So it takes a similar, so in this situation, we took the human SOD1, we took the primary sequence for the dog, and we put them together, and the computer predicted what the dog one would look like, called homology modeling.
What's the primary sequence? Do you remember? It's the amino acid sequence. So don't forget your levels of structure that we've already done.
So that's what I did. I actually this is actually printed out. This is the dog version of SOD1. So I went in, I put it in, I figured it out. It came back and told me this is it. I downloaded it. I 3D printed it, and we're off and running. So if I go to File, New Pymol window, I know, right? This is what the enzyme, or this is what it looks like. And you can see that we have important cofactors in there, right? 2 of them that are showing up right there. These cofactors come in and help with the enzyme function. One of the things I'm going to show you right now is if I go to display sequence at position 40, right there, you can see right there, if I show side chain as a stick, you can see, and if I'm going to color that by element, you can see that I have an E, which is which amino acid? I've got 2 oxygen right there. A little longer. Actually, it's a little shorter, sorry. Shorter. It's aspartate, right? I know that was 2 weeks ago, but.
Also known as aspartic acid because of that double bond.
So you can see that this sticks out right there, right? So if I change that to and I'll show you how to mutagenize it. So that's the normal form, right? So and I don't know what's going on here. This is a very interesting problem to me. So if you change that 40, I can go up to Wizard, go to mutagenesis, and I can change it to a valine. Trying to get it to change. I don't know why it's not doing it. Mutate to a valine. That did not work. This is not my day for getting things to work today. Why it says mutate to a valine? Why is that?
If you go from glutamic acid, or sorry, aspartic acid to a valine, is that a conservative change or non-conservative? Non-conservative, why? It's what? Yeah, it's got a charge in valine does.
So Wizard mutagenesis. It's still showing me. Why is it still showing me? What am I doing now? I don't know why it's not doing it. Huh? Yep, that's the danger of doing this live.
So what are the cofactors that are giving it the optimal differences? Yeah, we have two negative ions that are sitting in there. And cofactors are usually stabilizing the enzyme, or like I said, they're changing that active site to allow something to come in and bind.
All right, something's not right. I'm not doing it right, but that's the general idea. And we can change that, and then you can actually, again, you can mutate it, and then you can show, if you have one of these diseases, you can, again, show it, and you can show the mutation. If I have a valine in there, what's going to be the major difference between those two? Huh? That's okay, you can try. Yeah, one's very polar and it has charge, right? And one is valine, very charged. So what you end up with is and this is counterintuitive to me, right? But if we have two dogs who have that mutation on the end here, right? If you actually look at this as a surface, you can see where it is. So I can actually find where that valine is. And I can find it, and it's right there. And so if I have that, what ends up happening is, it doesn't affect, is this at the site of the enzyme? No, I think we can all see the enzymatic site is right there, right? That's pretty understandable. This mutation is over here, and it doesn't affect the enzymatic site. What ends up happening actually is that this protein starts to aggregate. Kind of like what other diseases are protein aggregation diseases that we've talked about. Alzheimer's, your prion diseases, those are all protein aggregation diseases. So this is what happens. I don't understand that. It's counterintuitive to me because you're talking about a large charge. Valine should be pretty inconspicuous, right? It's an E40V mutation. There's some that have an E40K mutation, which is lysine. So
What do you think would happen with lysine? What's unique about lysine? Is it conservative or non-conservative? Well, let's
no,
say it's
aspartate.
aspartate. Aspartate to lysine. It's charged, but is it charged the same? It's opposite charge. Lysine is positively charged, aspartate is negatively charged. So lysine has this nitrogen group that's hanging out there, and that's what's holding that positive charge.
So you can see what happens is in these diseases, sometimes you get some that are related to the enzymatic function and some are not related to enzymatic function. And that's kind of what you're looking, that's the kind of the level of which kind of disease pathology takes. And you can actually show this in PyMol. I obviously failed today on both counts. Apologize. That's the nature of doing this up front in front of you guys. Yeah. Oh yeah, good call. So if I'm going to treat this disease, do I want to attack the drug with the active site? So I'll just tell our story, right? So we started looking into this. I probably told you this before, but we started looking into this disease, and we're like, well, let's find a therapeutic, because my initial reaction was, hey, the enzyme isn't functioning, right? What does the enzyme do? Superoxide dismutase. It takes off free radicals. You've probably heard a ton about free radicals and your all the antioxidants, right? You've heard a ton about antioxidants, diets.
We thought we would just feed Cindy a bunch of blueberries, but then we thought maybe that wasn't the best plan we ever had.
Well, then, so I looked into it, and there's a vet in Missouri who's giving an siRNA silencing drug for this indication.
So is siRNA silencing going to interrupt transcription or translation? With translation. Remember,
You had a 50-50 chance.
The mRNA. Yep, it hits after the mRNA. That's where RNAi comes in. And so it's going to hit translation. So it hits before the protein is formed.
Yep. So how are they treating this disease then?
If they're using RNAi, what are they doing? They're stopping.
This enzyme goes away.
Yep, they're stopping the. Yeah, it's less. It gets minimized.
What would be the consequence of that, though?
Oxygen that are big
Two things, right? Great structure. Is that or not? It's very similar. What that is. Pretty. All. So I do complex and your foundation for diseases. A lot of your new diseases are immune. Your immune system back. Those proteins are they're normal. They have radical come together too. Side, right? So they want to good, right? there are going to be off. We we know what's on your you know process. You have right. I how to disease.
Technically, no, but practically, yes. Think of it as the same thing. Yeah, RNAi is typically the therapeutic. siRNA is what you use to do the therapeutic, and siRNA is the naturally formed molecule. Okay? Yeah. Okay. You guys ready to soldier on? All right. So the next fundamental concept that you need to understand is the catalytic triad. So the catalytic triad includes an acid, a base, and a nucleophile. And they always include an acid, a base, and a nucleophile. Now the catalytic triad is really kind of, think of it as the center of the enzyme's activity. In order to get that center to work, you must have these three elements: acid, base, nucleophile. So here are a couple common catalytic triads. So chymotrypsin, a very important enzyme here, its catalytic triad is serine, histidine, and aspartic acid. What is the acid, what is the base, and what is the nucleophile? Can we make a guess? Yes, ASP is the acid. Okay, we got one down. Two more. Histidine 57 is the base. Do you guys think so? Okay, so then that means serine is the nucleophile. Ben, are we right? Yep, you guys are right.
Chymotrypsin is a protease. That's primarily what you see catalytic triad. It's basically going to cleave
Proteins.
So when you eat something, it puts is what's going
It's going to cut up the different proteins. So you actually produce a lot of chymotrypsin in order to cleave and cut apart different proteins. So again, you should be able to look at the residues and identify what is the acid, what is the base, and what is the nucleophile.
And there is a question on the final on the catalytic triad.
Yes? Oh, am I just looking at the no, I'm not just looking at the shapes. What I'm looking at is what do we know about how these different amino acids can function? And once we know about their charges and we know about their pKa's, we know if they're going to function as the acid, the base, or the nucleophile.
What is aspartate? That's the name of aspartate.
Yep, and so that's how you know.
How many acids fit in that role? How many amino acids too? What are they? Yes.
Yep, glutamic acid. What's the single letter for glutamic acid? Good. So here's E.
We now know, yeah, exactly right. So there's the other one, right?
So here's your acid.
Yep. So histidine, what's the charge on histidine?
Is there a charge on histidine?
Positive. So what are the other two? What are
You
the
can
other-
look. That's fine to look.
Yeah, I'm not. We're not testing you over that
No.
right now. He's gonna have to. We're beyond that, so you just have to. So what are the other things that are in the same charge category? That. What are the other basic?
Residues, basic amino acids. From your chart, what else is basic?
Yep.
Okay,
So we get there. And then serine has what as a functional group?
here's serine. What's the functional group on serine? OH, right? So it's going to be a nucleophile. So these are going to be catalytic triads. They have a certain structure. These 3 amino acids have to interact in a very specific way, and you're going to see that here soon. And you should be able to identify what is the acid, what is the base, and what is the nucleophile from your catalytic triad.
The good news for you guys is we're not going to spend half a class period discussing the 7-step process in organic chemistry. We're grateful for this too, by the way, of how the mechanism of action of chemicals.
Actually, I was.
Really?
It's in here.
Okay.
Wait a minute. We're going to go through it fast.
Fine.
We're going to look at it,
Not
but,
much.
well, 3/4. No.
Half an hour.
Okay. So there are 4 main types of catalytic reactions. You should know these 4 types. There's an acid-base catalysis, where you're going to exchange protons. You're giving and taking protons. There's a covalent catalysis, where you're actually changing the reaction path. There's a middle ion catalysis, where you're using a redox cofactor. So that's where you're going to use some of those metallic ion cofactors. You're going to use that ion. And this is going to shift your pKa. What does that do if you shift your pKa? It changes the way it interacts with pH. It changes when it has a charge and when it doesn't have a charge, when it's protonated and deprotonated. So that's a big one. And then electrostatic catalysis. These give preferential interactions with the transition state. So remember, the transition state is the state of the molecules at the top of your activation energy. That's your transition state. So it's kind of that intermediate between your reactants and your products. That's considered your transition state. So an electrostatic catalysis is going to make that transition state more favorable. So we're going to look at each one of these in a little bit more detail, and I'm going to give you some examples of each of them that are going to be very central to what we're learning. Maybe not today, but eventually. So again, acid-base catalysis, lactate dehydrogenase is a common example of that. You can see here's your cofactor, remember, NAD+. We see that we've got an NADH here, we've got a free proton here, and so these are going to be oxidoreductase enzymes. And oftentimes, you've got histidine in the catalytic triad, because it can act as both a proton acceptor and a proton donor, and you have this proton here. So lactate dehydrogenase. Lactate dehydrogenase will come back to haunt you when we get to some of our other pathways.
Does anyone know what lactate is famous for? Does anyone know about lactate?
Yeah, and your muscles burn.
That's lactic acid buildup, right? So this is the enzyme that actually converts that you're using in energy when you don't have oxygen, you can't breathe, or it's not getting enough oxygen. So this is actually oxidoreductase as a cofactor. NAD+ is commonly the cofactor for a lot of the enzymes that you're going to see. This is actually backwards because
It's producing NAD+.
producing NAD+, that's what's using to get energy for you. So it's a little bit backwards than what you normally see. That's exactly what the kind of cofactor. It's not a cofactor there. It's actually
For the no.
So it's a little bit backwards from what you normally see.
So upstream of this reaction is glucose. We did. We talked about redox. We call them redox reactions, are very common. So you're going to involve some kind of oxygen. You're going to reduce that oxygen.
You transfer electrons.
Yep. It's not necessarily.
Sometimes.
Yeah, I mean, that's why this is
So
called an
they're related,
acid-base
but they're
catalysis.
They're not necessarily the same thing. Because that can be an ionic reaction too, right? That's not necessarily an oxidation reaction. You take hydrochloric acid, right? That's charges over electrons. That's not necessarily an oxidation reaction. That's an ionic reaction.
But that is also acid-base, potentially.
So sometimes they can be related, often they're separate.
So know lactate dehydrogenase, know that it's an example of an acid-base catalysis, and know that histidine is often found in the active site. So in covalent catalysis, this is acetylcholine esterase. Down here, you can see here's our active site channel that fits kind of right through here. And in this particular reaction, you have water that's attacking your carbonyl right here, and your oxygen, or you've got electrons leaving here. And so this gives you, this is your intermediate or your transition state, and here is your product. And so that allows serine to actually leave that reaction, and you see serine over here. And that produces acetyl-CoA. So metal ion catalysis, this was like my epiphany today, you guys. I was super excited about this. Apparently, I was more excited than others. But so about a third of all enzymes use some kind of metal ion. They need metal ions. In PCR, our polymerase has to have magnesium, or it won't work. Now, we hadn't really talked about that before, but there are a lot of enzymes that need some kind of metal ion for their catalytic function.
This is one of the reasons why if you suffer from migraines, taking magnesium
Will help.
A very common cure for some migraines.
Okay, so this middle ion is going to participate in the catalytic process. Here we have zinc right here in the center, and it binds to the substrates and orients them in the right way. So again, this has electrostatic stabilization, which shields the negative charges. And this is, again, another redox type reaction. So redox reactions are not always acid-base reactions. And it has this reversible middle ion oxidation exchange. So here's carbonic anhydrase with the zinc coordinating ion in the middle. So where's your catalytic triad here? It's probably got something to do with this histidine. There isn't one on this one, but it's got something to do with histidine. And you've got the cysteines.
Those are also very common in the catalytic.
And the cysteines, instead of forming a disulfide link, are being linked through the zinc, which is pretty interesting. So electrostatic catalysis, this is a particularly interesting one. Here you see cortismate mutase over here. Here's the other structure. And then here is your catalytic triad. And essentially, it's going to exclude water from the active site. So as the molecule moves in, it excludes water from the enzyme. And then because of the proximity of interactions, it allows changes in those pKa's of the amino acid side chains, and it allows bonding. So you're lowering the activation energy by favoring the transition state. You're allowing that transition state to be achieved easier and maintained easier. And you can see that because if you have the enzyme that sits kind of right in the middle here, it's stabilized by all of these bonds. That stabilizes that transition state. So it lowers the energy of the transition state, which lowers the activation energy. So now let's talk about how we name enzymes. There's a very specific way that we name enzymes. So the first part of the name is the name of the substrate for the enzyme. And the second part tells you the type of reaction catalyzed. And so it usually ends in ASE. So lactate dehydrogenase, lactate tells you the substrate, dehydrogenase, ACE tells you it's an enzyme, and it's doing a dehydrogenase reaction. So we have some trivial names. There are a couple exceptions that exist. Pepsin and trypsin are going to be some of your exceptions. But you can see you've got lipase, maltase, sucrase, lactase, and amylase. These are slightly different because the first part of it doesn't necessarily tell you the substrate. So these don't quite conform. These are more common names. They also have a more scientific name that, again, conforms to the rules and regulations.
Someone's very clever, so like lipase, not your lip.
Yeah. Somebody was, I think it was a lazy graduate student that said, I'm tired of saying lipid dehydrogenase. Okay, so enzymes are classified into 6 different groups. And this is according to the type of reaction that they are producing. So a redox reaction, which again is going to transfer this, is class 1. Your oxidoreductases, and that falls dehydrogenases, hydrogenases, oxidases, and hydroxylases fall within this category of redox reactions. So that is a class 1 enzyme. You can do a single replacement. So you've got A to B plus C, and B transfers between A and C. That's a transferase. You're just transferring a different component. So that's a transferase. That's a class 2 enzyme. And you can see examples underneath here. So your aminotransferases are transferases. Where did we see an aminotransferase before? Aminoacyl tRNA transferase. It falls in this category here. Yep. Then you have hydrolases. That's class 3. Hydrolases are a double replacement acid base. So you're just going to flip-flop. A was bound to B, now it's going to be bound to C. So you just flip-flop the two entities. You've got liases, and these are going to do a decomposition, so it just takes one thing and it breaks them apart. These are going to be like your decarboxylases, your aldolases, and your synthetases. You have isomerases. Isomerization just changes the specific isomer that B looks like. So it just changes one of the elements a little bit. So again, you're going to see a lot of isomerases. Some of these are going to be called mutases, which just means it's changing what that B looks like. And we're going to see these when we talk about some of our catalysis or our, and it uses a mutase. Then you have your synthesis, so A plus B equals AB, and those are your ligases. And we saw ligases when we talked about how to make plasmids, right? We talked about restriction enzymes and how we were going to paste things back together. Those are your ligases. And then these are examples of each one of those. So you should know the different classes of enzymes. So if I gave you an enzyme number 1.2.5.6, you should be able to tell me that's an oxidoreductase without knowing anything else about the enzyme. You don't have to know anything else about it. If you know that first number and you know these different classifications, you know what class of enzyme that is. That's the level at which I would like you to know this. So all enzymes are then assigned an EC number. So again, cytochrome CYP3A4 is a 1.14.14.1. So the first digit tells you the class. So a 1 is going to be what class? Oxidoreductase. Yep, good. So this is an oxidoreductase. And we know that based on the fact that it does this oxidation reduction reaction. And then the second is the subclass. The third is the sub-subclass. And they describe the reaction. And the fourth tells you about different enzymes of the same class that function kind of the same. I don't expect you to know anything beyond the first digit. Just kind of know those general classes, those 6 classes. So an alcohol NAD plus oxidoreductase is 1.1.1.1. That was the very first one. Again, the classification doesn't take into account the amino acid sequence or the homology, the protein structure, or the chemical mechanism. It only looks at this particular classification, and so they're not necessarily related beyond the fact that you have 6 different groupings, and you should know that if I tell you that it's in group 3, you should be able to tell me, oh, that's a double replacement acid base, so that's a hydrolysis. So an alcohol NAD plus oxidoreductase, 1.1.1.1, what kind of enzyme is that? From those general classifications. It's a class 1, and what's a class 1? Oxidoreductase. It even says it right in the name here. So here's just another way to look at that, because some people that doesn't really mesh with the way that they think about things. So this is just, again, another way to look at the same 6 classes. Some people see it better one way, some people see it better the other way. So I gave you both. So class 1 oxidoreductases, these catalyze the transfer of hydrogen or oxygen atoms or electrons from one substrate to another. So here is our CYP3A4. You can see here's our endoplasmic reticulum, which is where CYP3A4 is found. And this is going to be an oxidoreductase. So these are considered redox reactions. There's an electron donor and acceptor to complete that reaction. So you can see how that would happen here. Here's our NADPH, NADP. So this is going to be an electron receptor, and you end up seeing the quinine, which we'll talk about, and hydroxyquinone. Our transferases catalyze a group of transfer reactions. So here's one particular example with aminoacyl tRNA transferase. Here's your amino acid. You have your protein, your N term, your C term here, and then when you put in the amino-acetyltRNA tRNA transferase, it transfers the amino acid to the N terminal to the growing chain. Here's an additional example down here where you've got glutamate and an alpha-keto acid, and then you end up getting alpha-ketoglutarate and an amino acid. This is how we produce certain amino acids. So again, just understand that this is, you're transferring oxygen or hydrogen. Sorry, that's class 1. You're transferring anything else, essentially. So hydrolases, in this particular class, these are going to catalyze hydrolytic reactions. This one that we're looking at right now is ubiquitin carboxy terminal hydrolase L1. And these have this type of general form. So the catalytic triad contains a cysteine, which we talked about before, that cysteine is common. So it's got a cysteine at position 90, and an aspartate at position 176, and a histidine at position 161. Which one is the acid, which one is the base, and which one is the nucleophile? So you have cysteine, you've got aspartate and histidine. Is aspartate the base? Aspartate is the acid, yep, because it's also known as aspartic acid. So now you have to decide between cysteine and histidine for which one is the base. Histidine's the base, so cysteine is going to be which part? The nucleophile, yes. So that's your catalytic triad. And ubiquitin is basically the molecule that gets added to proteins to tag it for destruction. If you have an ubiquitin on you, it means you shouldn't be here anymore, and you get tagged for destruction. Did you have? Okay. So then we've got our lyases. Now these are non-hydrolytic because hydrolytic are from enzyme classification 3. So this again is going to remove functional groups from a substrate. Here you have your citrate. You've got APT, ATP, citrate, lyase, if I could speak. And that turns into oxaloacetate. And then you can add that to acetyl-CoA, which gives you steroids and fatty acids. And this is your enzyme right here. So this includes your decarboxylases and your aldolases. And it includes synthesases in the addition direction. So your lyases are kind of a weird classification because they can include other classifications. You've got your isomerases here. So again, they include isomerization. Do you guys remember racemic mixtures? Where you have, yeah? Okay, what are racemic mixtures? Yeah, so you can have lots of different kinds of racemic mixtures. So you can have a mixture of D form and an L form. You can have a mixture of chiral and an L and R form. So you can have different mixtures of the same compound, but they have different chirality. They have slight differences. So racemic doesn't actually tell you anything about which one is favored more than the other. You could say that you have a 50-50 racemic mixture and then they're both equally represented, but it doesn't necessarily have to be that way. So you can see we've got an isomerase right here because glucose 6-phosphate and fructose 6-phosphate are going to be the same molecule, but slightly different. So you've got an isomerase here. You've got another isomerase down here between glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. This is another isomerase. So this is slightly changing that particular substrate. A lot of times you've got cis-tran isomerizations. So remember from chemistry, Cis means your functional groups are on the same side. Trans means they're on opposite sides. So a lot of times that's what you end up doing here. Of a double bond. Yes, thank you. Then you have ligases. Again, they're going to catalyze the synthesis of a lot of bonds, and they're coupled with a breakdown of energy containing substrates. So they take energy to make those bonds, but they're going to form bonds. We hear about those with DNA ligase a lot. Again, depending upon the source, they're going to require either ATP or NAD+ as cofactors. Do you have this one to do?
No, I'm fine with it.
Okay.
Let's, what I can do, I'll do it, do I get to do that? Do it. Let's keep it.
We all agree. It's fine. Oh, I'm sorry. Sorry. I forgot I was wearing a mic. I don't know what happened there. Okay, so a lot of the mechanisms of action are based on proximity, right? Things get in proximity and stuff happens. So that's called the proximity effect. And you can see that we end up getting certain intermolecular reactions that happen a lot faster than the intermolecular interactions. And that's just simply because of proximity. The one kind of orientation effect that you need to be aware of is the steric crowding, which could produce a rate enhancement because you could get to the transition state and then immediately go on to the product state because of steric hindrance. And so you can get a rate acceleration because of that. And that, again, that's the orientation effect. So the one that we have talked a little bit about, but now I'm going to show you exactly how this can happen, is this transition state bonding effect. So we're going to stabilize the transition state now so that we can actually get to the transition state. Now remember, everything wants to be at its lowest energy level. Everybody wants to lay on the couch for as long as possible without getting up. So we need to lower the energy level of the transition state. One of the ways to do that is to stabilize it, because remember, everything is going to find that lowest energy level. So we want to make that energy level even lower by allowing our enzyme to bind to the transition state and minimize it. So here we've got no enzyme. You've got a substrate, which is your middle stick. The transition state is the bent one, and then the product is the broken one. So you have this large energy of activation to get to a bent middle stick, right? It takes a lot of energy for you to get there and to maintain that. So if we add an enzyme, like a magnet, for example, this is an enzyme that's complementary to the substrate. It's going to keep it in the substrate form. If the magnet is complementary to the substrate, it keeps it in the substrate form. It prevents it from going to a transition state. But if the enzyme looks more like the transition state, then this substrate wants to be in the transition state, because now it's surrounded by this area that's not full, and it doesn't fit quite right. And so it wants to have that fit. And we're, so they're ionic bonds, yep. There are hydrogen bonds. There are van der Waals interactions. There's covalent bonds. What do you guys think? Mm-hmm. You actually share electrons. They're the hardest ones to break. So would you want something that's really hard to break for your transition state? No, because you want it to go to the product. So you wouldn't want something that's so stable that it has to maintain the transition state. So we can mark covalent off the list. So
It's gonna be all the other ones, all the other non-covalent interactions, including hydrogen, are gonna be what's gonna stabilize that.
So when we talk about some of the enzyme classifications where we look at electrostatic interactions, those are going to be more ionic type bonds. So these could be ionic. So what ends up happening, if you have the enzyme that complements the substrate, then you actually lower the energy of the substrate, and this is the one time that you change your overall Gibbs free energy, because you have changed the energy of the substrate to start. So then in this case, you've changed Gibbs free energy. In this case, you have changed the energy of activation, because you have taken your transition state, listed as double dagger here, and you have lowered the energy that it takes to arrive at that state and maintain that state. So you've lowered that energy here. And so you've changed your activation energy, but your substrate and your product are still the same, so you have not changed Gibbs free energy. Remember, delta G is Gibbs free energy. It only relies on the reactants and the products, and then delta G double dagger is activation energy. That's what's going to be affected by your enzyme. So here's another way to look at that. Here's a transition state bonding. Here you have your particular molecule. You've got your enzyme. That molecule goes into your enzyme's active site. Here's your aspartic acid, your glutamic acid right here. And you end up having this reaction that occurs. You add water in, and you can actually get the reaction. So we saw with our metal rod example that we started with a substrate that was a straight rod, and our transition state was a bent rod. So if we use our enzyme that looks like a bent rod, and then the substrate bends after that enzyme comes into contact, that's an induced fit model. So in an induced fit model, your enzyme looks like one thing, and when your substrate goes in, it changes its shape to conform to that enzyme. Or, alternatively, the enzyme changes its shape to conform to the substrate. But one of them is changing its shape. So that's going to be an induced fit. Here's our active site of our enzyme. You can see that sitting right here. Here's our substrate that fits right in there, and then our enzyme closes around it to induce that fit even further. So this induced fit system is a way a lot of our drugs actually go in and can block these enzymes, because all it needs to do is mimic the shape of the substrate. It doesn't have to really mimic the chemistry of it, only the shape. And that's how you can block that enzyme function. Second mechanism of action that we need to talk about is a lock and key mechanism. In a lock and key mechanism, your key is the substrate, your lock is the enzyme, and you complex them. So the key fits into the lock doesn't change to fit the shape of the key does not change its shape to fit the lock. They both have the proper shape to start. And if they don't have the proper shape, they don't interact. So this is a lock and key mechanism. So here is an example of that. Here's our key active site. It's amino acid residues, and here's our substrate that's going to come and fit right in here. And you guys have seen lots of examples of that. So here's chymotrypsin. Again, it's a proteolytic enzyme. This is secreted by your pancreas. It does a lot of hydrolysis. And here is this primary structure, secondary structure, tertiary structure? What level of structure are we looking at here? Primary. Yep. It's primary because we know that we're looking at the amino acids. So chymotrypsin uses serine 195, and serine becomes the nucleophile. Serine is going to attack the carbonyl group of the protein substrate, and then you have, here's the first couple of steps, an acetylation reaction, and then a deacetylation reaction to release it. So you get an acyl enzyme as your transition state. So histidine removes the proton from serine 195 over here. So does that mean histidine is acting as the acid, the base, or the nucleophile? What's it acting as? Yep, and so your acid, your base, and your nucleophile. So aspartate, the acid, is going to orient the histidine so that it can be a better proton acceptor. So all of these have to work in conjunction, and they all orient each other by these non-covalent type interactions that they form with each other. This is another way of looking at it. It's important that you realize there's an oxyanion hole in the serine protease mechanism, and this is your oxyanion hole, which gives you an electrostatic catalysis. Now, the reason that we go through the mechanism is because the final does ask about this oxyanion hole and the transition state. Remember, it's an acyl transition state, and you have this oxyanion hole that fits up here. You've got serine, histidine, aspartic acid right over here, and that gives you your catalytic triad. And this, again, is a preferential binding to the transition state. So this is when the anoxion hole is filled, it gives you a tetrahedral intermediate. That's the part you have to know. All the rest of this, you should know that you have an acyl enzyme mixture, but that the intermediate is a tetrahedral and you have an anoxion hole. That's the part you have to know from the chymotrypsin mechanism. And that specificity, here's your oxyanion hole that's sitting right here. And that it's that specificity of how this actually looks that gives you the specificity for the transition state. So again, we can get an enzymatic substrate selectivity, where you can see here we've got no binding. Here we've got binding but no reaction. So we can just mimic the structure, and we can prevent the enzyme from functioning. A lot of enzymes aren't as specific for primary structure. They're more specific for secondary or tertiary structure.
That actually is the top reaction is the one.
This is the reaction
That is the reaction.
that should
What's on the
happen.
left?
Is this reactants or products?
What is that? What is on the top?
This is reactants. What's the molecule?
Well, no. What's the other? What's the ring there? You're in the right area. It's amino acid. Phenylalanine. Yes, it's phenylalanine hydroxylase. So hydroxylase is adding a. What's that? What's that on the product on the right? That is hydroxylase. That whole molecule. Yes, which is also known as tyrosine. Yes.
So this is how we convert.
Phenylalanine
I know, this is rough, guys.
to
You're going from phenylalanine to tyrosine.
you know why they
Do you remember
they're born?
right when babies are born, they take a heel prick and they take blood. What are they looking for? They're looking for metabolic syndromes. There's a very, very specific one, though. They're testing the blood. Yeah, it's not diabetes. It's a metabolic disease. Diabetes is. Not quite. Nope.
They're actually testing for this disease right here, which is you're missing that phenylalanine hydroxylase.
It's called PKU.
Phenylalanine.
ketourenia. Yeah. So it's PKU because if you're missing this enzyme, you can't make tyrosine. You can't process phenylalanine. If you can't process phenylalanine, you can't process protein. So that's a big one.
This actually shows that, you know, the different forms of alanine, B form of phenylalanine,
Mhm.
it's very specific, right? And then you can block it, right? This is how we use, what do we hijack? How do we hijack this mechanism, this selectivity mechanism? You guys are all pharmacists, right? That's exactly
Going to
how
be.
we hijack nature, is this selectivity.
We put a drug in here.
Reaction, right? So we're blocking based on a reaction. This is obviously not a great reaction to block, right?
You really don't want to block this
We don't
reaction.
Really want to block this, right? That would be a bad drug to develop, right? Since you would cause PKU, that's not a good thing to cause, right? But there are drugs that we want to block, right? That's exactly the mechanism that we take to solve that problem.
Which is why we can use computer modeling and homology modeling that Ben just talked about to predict what a drug should look like. And then once we know what it should look like, we go to these fantastic medicinal chemists and we say, make me a drug that looks like this. And then we go back to our lock and key and our induced fit. And as long as it can fit in there and it's close, or it fits in there and it's perfect, we can bind up that enzyme. Then that means that we don't have to do RNAi, we don't have to knock it down, we don't have to mess with genetics. We can just do it this way.
You'll want to know that a lock and key and an induced fit. You'll want to know that induced fit has allosteric changes.
We haven't gotten there yet. We will tomorrow. I think so, because we're talking about enzyme kinetics. So how do we stop an enzyme? We just talked about a couple different ways to stop it. But remember, we can alter a protein's shape through forms of external stress by applying heat or changing the pH. We can see this when we talk about an egg. An egg is protein, right? We change the form of the proteins of an egg by applying heat. It changes the form. It makes that really runny stuff become white. You're changing and denaturing that protein. So remember that a denatured protein can't carry out its cellular function typically. And this is exactly what bacteria do. When we give specific drugs, bacteria have developed enzymes to change those drugs and make them not as good. So they've altered some of their enzymes. These are all different ways that bacteria have gotten around a lot of our antibiotics. But essentially, this is what it comes down to. So in one case, you have an enzyme here. The bacteria produced an enzyme. We made a drug to block the enzyme. So the bacteria altered the enzyme so that it cannot accept the drug anymore, so that the antibiotic does not bind anymore. It makes it more selective. The enzyme still functions the way the bacteria needs the enzyme to function, but it can no longer bind the antibiotic that we made. So it actually changed the enzyme. Sorry. Yes, it happens through mutations that changes the enzyme, and that gives a selective advantage, and so then that trait gets passed along. That's how resistance happens so quickly. So in this particular case, the enzyme actually destroys the antibiotic and prevents it from binding to its target site. So here's our cell wall. Here's our antibiotic. It would normally fit in there. In one case, the antibiotic is destroyed. In the other case, the target site, and it goes out and it alters the antibiotic itself. So it produces an enzyme that's going to alter the antibiotic.
Does anyone know what the most famous antibiotic destroying enzyme is?
It's not the Death Star. I'll let you know that.
An enzyme. So the enzyme is actually
So it's coming out here.
Yeah, the DD carboxylate, what is the
Oh, man, you would ask me. I don't remember. It's a DD alanine, and it prevents it. I gotta look it up. It's in one of the early lectures.
But I feel that as a side note, it just kind of gives you to understand that or bacteria can use enzymes also as a weapon against our drugs. So we use drugs as a weapon against their enzymes. They develop enzymes to take out our drugs.
It's kind of an arms race. It's pretty cool, actually. So remember, we can also have enzyme antagonists. So here's our natural chemical. Here's our receptor site. This is before a drug. The chemical fits in here, but it's not an exact fit. So one of the things that we can do with an agonist, which is going to help the enzyme work, we can use an agonist drug that's going to fit into the receptor site and then better fit the natural chemical. It comes in and sits in the agonist. Or we can develop antagonists. Antagonists are going to act as little blocks for the receptor site and prevent the drug from, or the natural chemical from entering the receptor site, so we can block that activity as well. So remember the difference between agonists and antagonists. That is the end of today. Well, probably not the end of your day or mine, but it's the end of lecture today. Any questions? Sure. An agonist is going to be something that helps the drug act better, and an antagonist is going to prevent the drug action. Or it's a drug that's going to prevent the natural chemical from acting. So agonists help, antagonists prevent.
We use this term all the time when we're talking about drugs. I don't know as pharmacists you will, but
You will. All right, so tomorrow we're going to dive back into kinetics, and we're going to talk specifically about enzyme kinetics, which will be pretty fun. Ben hates this lecture. I think it's pretty cool. So we'll see you guys tomorrow. You do. You tell me all the time.